Okay, let's unpack this. Imagine you're a clinician, patient's got a severe infection, things are moving fast. You're under immense pressure, right? But the diagnostics, they often feel like getting puzzle pieces one by one, sometimes days apart. It causes these awful delays, loads of uncertainty.
Yeah, and what's really fascinating or maybe frustrating is how those traditional tests, a cultures, maybe a targeted PCR here, an antigen test there, they just give you bits of the picture.
Right, not the whole thing at once.
Exactly,
Wow.
especially not in those first crucial hours, and you're sending samples off maybe to different labs even, just waiting.
Time you don't have.
Yeah.
So clinical metagenomics then, the idea of looking at all the microbial genetic material in one go.
Mm.
Our deep dive today, it's really about a study proposing a unified way to do just that, to cut through those diagnostic headaches. And the sort of surprising key seems to be this clever way they clean up the sample first
Yeah, get rid of the noise.
to get you the info you need much, much faster.
Right.
So this study really puts its finger on a big hurdle, doesn't it? That current metagenomic methods aren't great at getting both RNA and DNA microbes at the same time,
Right, or they make things more complicated, splitting samples, adding steps.
which adds time.
Exactly. So this research, it details a pretty rapid strategy. It's focused on getting rid of the host DNA, basically clearing out the patient's own genetic material that swamps everything.
How do they do that?
First, they spin the sample down centrifugation to pellet the human cells. Then, they use these tiny zirconium silicate beads, about 1.4 millimeter, to physically break open only those human cells.
Okay, so the human DNA and RNA spill out.
Precisely. And here's the really neat part. They add a specific enzyme, a nuclease. It's called HL-SAN nuclease from ArcticZymes Technologies.
HLS-SAN.
Yeah, HL-SAN nuclease. Think of it as being programmed to just chew up all that free-floating human genetic stuff that's been released.
So it specifically targets the free human DNA and RNA.
That's the key. It goes after the loose stuff. Now it's a bit less efficient on RNA, maybe 10 times less than DNA, but its main job is clearing out the host material.
And crucially, it doesn't mess with the DNA or RNA still inside the actual microbes, the bacteria or viruses you're looking for.
Exactly right. The data suggests it leaves the nucleic acid inside intact microorganisms completely alone because those microbes haven't been broken open yet.
That sounds like a huge advantage.
It is, and the depletion is pretty effective. They measured a median reduction in human DNA of about 8 seed values.
8 CD values, so that's like, what, roughly 250, 260-fold less human DNA?
Yeah, around 256-fold reduction. That's a massive decrease in background noise.
Wow. Okay, so that must seriously boost your ability to actually see the microbial signals then? What does it mean for the range of bugs you can detect?
It opens things up considerably. This way you can simultaneously detect a really broad range RNA viruses like flu, DNA viruses, loads of bacteria.
Even the tricky ones.
Yeah, including atypical ones like Legionella, Chlamydia, Mycoplasma, and fungi too, like Candida or Aspergillus, all in the same run. They showed detection of influenza A down to, I think, 70 copies per microliter, and Staph aureus at 10 to the 3 CFU ml.
Okay, broad detection, good sensitivity. What about the clock? You mentioned speed earlier. Is this genuinely faster for the clinician?
Oh, definitely. That's maybe the biggest win. The whole process, sample into final report out, can be done in just 7 hours.
7 hours end to end.
Mm-hmm. And get this, they can generate preliminary reports automatically after just 30 minutes of the sequencing run starting.
Half an hour for a first look.
Yeah, enough to potentially guide initial therapy. And the full comprehensive report, which includes things like antimicrobial resistance genes they might find, that's ready within 2 hours of sequencing starting.
That really does sound like it could slash that diagnostic waiting time.
It tackles that bottleneck head on.
So a single workflow, fast results, broad detection. It sounds, well, almost tailor-made for a busy clinical microbiology lab.
That's certainly the goal, yeah.
Yeah.
It's designed to be clinically deployable, a unified approach instead of juggling multiple different tests. Now, obviously, for widespread use, you'd need more automation, standardized controls, things like that. The study acknowledges that.
Sure, implementation details.
But the core advantage over the current sort of staggered testing approach is pretty clear. And it's not just about what pathogen it is, you're getting genomic data too. That can tell you about potential drug resistance or even help track an outbreak much faster.
So this shows quite a leap forward, potentially, moving away from piecing things together towards one comprehensive, rapid snapshot of an infection.
It certainly seems that way. And connecting this to the bigger picture, it leaves you with a really interesting question to think about, doesn't it?
Hmm.
If we get really good at this rapidly identifying everything in a sample, even novel or completely unexpected pathogens, how is that going to fundamentally change treatment decisions? And not just for one patient, but for public health surveillance? How does it change our preparedness for whatever comes next?
That's definitely something to mull over as we think about where clinical microbiology is headed.